All right, so here we are, ready for, this should say test 3, because test 3 is our muscular system, not test 4. Sorry about that. Here we go, fix that. So far you guys have covered a lot of ground in a short period of time. This is a really intense way to do this class. If you're still sticking in there, and some of you have gotten pretty decent grades going, which is really difficult to do in this format. So I applaud you for that. I know that there are a lot of people dropping the course by this time because they just don't have a chance at passing it. You do need to successfully complete this to be able to do 1552 in whatever format you want to do it. You're probably figuring out that doing a full online is probably the hardest way to study anatomy, and particularly when you don't have a lab, you don't have that lab time. And in 12 weeks, that's where I was going. Okay, so let's just kind of go through the notes pretty quickly. The idea here isn't to just read them to you. It's just basically to point out some important things and to kind of give you some tips on how to prepare, right? So when we get into this first section, what you're seeing here is how your muscles are built, bundles of bundles of bundles, and that each of these bundles, you can see these white lines in between here, those are those are connective tissue layers. So we need to understand the connective tissue layers as well as the bundles. So what I recommend you do, besides looking at many images, this is one, but it's similar but slightly different what's in the lab manual, because the resource I used for this wasn't Wiley. So it's the same, but it looks a little different. So make sure you look at it there too. Our textbook also has, I think it's exactly the same as what's in the lab manual. So use this. You can see that there are 3 layers of connective tissue. The epimysium surrounds the entire muscle. The perimysium is around each fascicle, and the endomysium is around each muscle cell, which is a muscle fiber. So EPE, kind of see how that works? Epi, peri, endo. Endo is always your deepest layer of anything. So that's kind of easy to get that. That's the furthest one in. And then if you're wondering which order the others go, EPE, it's like the P's in the middle. So maybe that'll help you with that. I'm not sure. This is just getting into the cellular makeup of muscle cells, understanding that they are actually, a muscle fiber is a muscle cell. And these are some of the longest cells that you have in your body. You've got muscle cells that go all the way from your butt cheek, basically, to below your knee. That's your hamstring muscle. So the muscle fiber extends almost that entire length. That means that there are cells that are that long, right? So that's kind of impressive. And since these are some of the largest cells in the human body, and they're made mostly of protein, and we know that protein comes from this process called protein synthesis. In order to do protein synthesis, you've got to get DNA from the nucleus, a copy of it, right? Transcription, you remember this from biology class? Since the cell is so big, we have to have many nuclei along the way. And the other thing we have a lot here are many mitochondria, because muscle cells produce more, they use more energy, and therefore they need to do more cellular respiration, which is what the mitochondria are for, right? So we've got that too. All right. When we break it down, we see that the myofibril, that's the smallest, that's an organelle, the smallest grouping of bundles within the cell, right? So this is an actual, within a muscle cell, we've got these long structures called myofibrils, and they're made up of a smaller structure called the sarcomere. And each sarcomere is end to end to end. Kind of like, if I were to stack these whiteboard markers, this would be one sarcomere, this would be another one, then if I had another one and another one, and this goes all the way down the length of the myofibril. And it's these units that are actually shortening when the muscle contracts. So the myofibrils are what are shortening. And as a result, The sarcomeres are shortening, therefore the myofibrils shorten. The myofibrils extend the whole length of the muscle, so the whole muscle shortens. Okay, that's how it works. So there's a couple good flow charts here. This is good. Feel free to use these, redo them, whatever works best for you. But basically what you've done here by using this sort of a format is you can condense down like 10 slides or more into just this. That's what you should be doing. And if you're not actually taking notes on this stuff and then rewriting those notes into a more concise format to study, you're probably struggling because you can't study these slides. That's not what they're for. These are to deliver the information to you to supplement what you're getting from your lab manual and your book. But really, this is like where most of the information is coming right out of these slides. For your test, you probably figured that out already. Okay, so this one also is just another way of looking at all the things that make up a muscle cell. All right, and anytime you're going through these slides and you see something like, oh, I need more on this cytoplasmic reticulum. Okay, fine. Go into the textbook, because there's a lot more detail there, and you can fill in any kind of spaces that you have. Be sure you're fully understanding everything, but it's more than understanding. It's more than just, oh, I get it. You got to be able to reproduce it, right, on a test. And yeah, I know our tests are all restricted response, multiple choice. That makes them a lot easier than if you had to write all the answers out. But it doesn't make it easy to do well on it. There's so much material. All right, next part is we break down the sarcomeres, again reminding us about myofibrils, that they're made of sarcomeres arranged end to end. And this is where the contraction happens. And basically, this next section just basically goes through all the different parts of this, talking about thin filaments, and those are here, these purple ones, and that they attach to these Z lines, right? The Z lines anchor them down. And then the thick filaments, those are the dark magenta colored ones here, that don't attach, but they're still anchored to the sarcomere right in the middle. There's a little protein, think of this as like a thumbtack holding it in the middle of the sarcomere. All right? This is really useful to learn. I use a little acronym, Zoom in a Hot Mustang, to help you just kind of remember all the different letters, like Z is Z line, and look at where they are. So one good exercise for this is what would look like under a microscope here, and this is actually what that corresponds to. So be sure that you understand what each of these ZIAHM things represent, and you can find it all in this graphic. But also, you should be able to think through what's going to happen to each of these zones and regions when the muscle contracts. Because when muscles contract, these thin and thick filaments that you see up in there, they slide together like this. So the thin filaments never change their length. The thick filaments never change their length. They just slide together over each other. And that pulls the Z lines at the borders of the sarcomere towards the middle. And if that's happening all the way down the muscle, the whole muscle is going to shorten. Right? So that's basically what we got going on. The next section is just goes into the thick and thin filaments. This is showing you here, these little myosin heads are trying to attach to the thin filament and pull it towards the center. That's how it works. So they call it the sliding filament theory. Okay. First thing that we need is a signal from a nerve. So your brain sends a signal down through the nerve. We haven't studied nerves yet. That's coming. That's coming soon. But then we'll learn how that signal is conveyed down the neuron. But for now, we want to get that this junction between the neuron and the muscle, the neuromuscular junction, that's really where the muscle gets its signal from the nervous system. And it happens just like any connection between neurons, except in this case it's connecting between a neuron and the muscle membrane instead of a neuron to a neuron. So we have the signal that comes down, And there's a neurotransmitter. You can go through all these slides to get that. And eventually we get to this idea of sliding filament, right? So what is it? And we went through these steps already. Muscle impulse. So what a muscle impulse is, let's just get this square. So when this electric signal hits the membrane of the muscle, we call the electrical signal coming through the nerve a nerve impulse. So that word impulse means it's an electrical signal being sent down the neuron. When it hits the muscle, instead of another neuron being there, it's the membrane of the muscle that conducts the signal. So since it's still electrical and it's being conducted through the muscle, it's now known as a muscle impulse. The impulse part's the electrical, and it's the membrane of the muscle that we're talking about here. Okay? All right? And this goes through all the steps. You might need to review, about the sarcoplasmic reticulum a little bit more, but that electrical signal is going to cause the sarcoplasmic reticulum to dump calcium out into the cytoplasm, which is called sarcoplasm of the muscle. And it's that calcium that really allows the contraction to happen. It's kind of the monitor. It's the thing that if it's present, the muscle shortens. If it's not present, the muscle relaxes. When it's present, it shortens. When it's not present, it relaxes. So it's calcium within the muscle that's really the motivating factor here. And that calcium was released originally because of the nerve signal that came down, right? That's what it's about. And that's why that sarcoplasmic reticulum is so important inside the muscle, because that's what stores and then releases calcium when it's told to. And then it recovers it, and then it releases again. Then it recovers it, and it's constantly going in and out of the sarcoplasmic reticulum. When it's out and in the cytoplasm, the muscle contracts. When it's in the sarcoplasmic reticulum, calcium can't influence the muscle contraction. So that's basically what we got going. Okay. All right. And this is basically just going through here showing the shortening of the muscle, right? And you can see how these fibers are all the same length the whole time. It's just that because they're drawn closer together, the borders of the sarcomere, the Z lines, are pulled closer towards each other, shortening. And if we have a bunch of these next to each other, the whole thing's going to get shorter. All right, ATP is a little bit confusing in how it works. So let me just give you the breakdown on how that works. ATP is used by the thick filament, the myosin filament, and it had these little cross bridges, extensions that are trying to connect to the thin filament, right? And when the calcium is present, the binding sites, the little places that it wants to connect to, those are opened up and available. So that's the whole idea what calcium's doing, is allowing this to connect and pull, right? Well, what ATP does, which is kind of interesting, ATP is needed for after this connects, ATP causes it to disconnect, which is kind of weird because we think, well, the pull part must use the energy. That's the ATP, right? It doesn't. What it does, the ATP causes the disconnect, and then the ATP breaks apart, liberating its energy. So as soon as ATP binds to the cross bridge, it separates it from the thin filament. Now the ATP splits into ADP and phosphate. As it splits, energy is released. See, the energy is stored in the bond between the phosphate and the molecule. When I break off a phosphate, energy is released, and that energy can be used by the cell. In this case, what the energy does is it causes that cross bridge. Think about a rubber band or a bow and arrow. When you use energy, you had to pull back the bow, right? But once you got it back there, you've really spent the energy. All you have to do is let go with your fingers, and the arrow flies away. The energy was spent pulling it back and getting it ready. That's what's happening here. So what's happening here is when the ATP splits and releases its energy, that energy takes the cross bridge, pulls it back, just like pulling back a bow. Now it's ready. if there's a binding site available on the actin, because calcium is present, right? We need a calcium present to open up those binding sites. Then it attaches and pulls. Now another ATP is necessary to break that. The ATP then breaks apart, gives its energy to the cross bridge, pulling it back to be ready again. Let's say the calcium's gone. Now this is covered up. Nothing happens. It's waiting. It's just ready to go. And as soon as a binding site opens, it pulls. Now I need ATP. See, the ATP energy is spent, and then it's just waiting there, all stored up, waiting until it can pull. So ATP is used in two ways in muscle contraction. One way is right here, and this is on the thick filament. This is a thick filament only that has the ATP, little receptors to bind to. And it's that energy that's used to recock. Think of like cocking a BB gun, like a Red Ryder BB gun from Christmas Story, right? You had to cock that, then you could shoot it, then you got to cock it again. Well, that's what's happening with the energy. It's recocking it, getting it ready, then it shoots, then we got to recock it again with another ATP, then it shoots. Okay. I mean, it is necessary to contract the muscle, but let's say there's no ATP. Let's say there's no ATP. Let's say that, and there's a few slides later, there's a condition called rigor mortis that can happen. Let's get to that here. What rigor mortis basically is, that's why they call dead bodies stiffs, right? It's because of this condition, and here's how it happens. After you die, the calcium leaks out of the sarcoplasmic reticulum into the muscle. Now what that calcium does is it opens up all these binding sites. And since the ATP energy has set up all these cross bridges, making them all ready, they connect and pull. But there's no more ATP. So without the ATP, you can't disconnect the cross bridge. So they stay there in this attached, sustained contraction. There's no way to get the calcium out of the sarcoplasm because that requires ATP, and there isn't any. So this is why it sits like that. It sits in this contracted position. That's rigor mortis. Now, eventually, the proteins, the actin and myosin, the two filaments are going to denature. They're going to decompose, basically, and that's going to allow it to relax. But that takes, depending on the conditions, how cool it is and all that kind of stuff, that could take, days for that to happen. So, medical examiners use this. You've probably watched plenty of medical shows where the state of rigor and all that kind of thing. That's what they're talking about. And they use it to estimate like time of death and all that, and based on if it's still in rigor or if the rigor's no longer there, that tells them how long ago and all that kind of thing. Okay. All right, so I said one place, and you can see it in there, is that the ATP breaks into ADP and phosphate, that pulls it back. Okay, now we can pull again if there's an open binding site. The other thing it's needed for is to remove the calcium from the sarcoplasm and put it back into the sarcoplasmic reticulum. You see, the thing is, in biology, we like to use diffusion anytime we can because it doesn't take any energy for diffusion to happen. So when the gate opens, the calcium gate opens, the calcium that's in a high concentration inside the sarcoplasmic reticulum, it can just leave. It just diffuses out till it's equal, right? It'll go to equilibrium. But in order to get it back in there, see, it won't move by itself up against a concentration. It's like a ball won't roll up a hill by itself. You need someone to push it. Someone needs to spend energy to get the ball up the hill. Same thing, we need to spend energy to get the calcium into the sarcoplasmic reticulum. So ATP is needed in order to do that. So without ATP, the calcium can't be removed and be ready for another contraction. Okay. All right, so that's pretty good. I don't think there's a whole lot else to talk about. You can look into some of this here, talking about summation and twitches. And this is about how each muscle fiber can generate more force if the frequency of stimulation is faster because of what I just said. So if I contract a single muscle fiber, that's called a twitch. After it contracts, it relaxes, right? And if there's enough time during that relaxation phase for all of the calcium to be pumped by ATP back into the sarcoplasmic reticulum, you'll see like the top image there. But let's say there was a stimulus, so some calcium was shot out there and the muscle contract, and the ATP started pushing the calcium back into the sarcoplasmic reticulum, but another stimulus comes before it all gets back in there. If another stimulus shows up before I could get all the calcium out, I'm going to get another shot of calcium released in there. So now I still have some from the last time, plus what was just sent in there, and that allows more binding sites to be opened than before. And if more binding sites are open, more cross bridges get to attach all at the same time and pull. And because we have more cross bridges pulling at the same time, more force can be generated by that fiber. And that's what the graph is showing, is the amount of force. So the faster that these stimuli are coming, the more calcium ends up building up inside the sarcoplasm, the cytoplasm of the muscle, right? And the more calcium we have out there, the more binding sites. The more binding sites, the more cross bridges engaged. The more cross bridges engaged, the more force. But there's a limit to it. If we're coming down at the maximum frequency, right? That's what we're seeing at the bottom there. Maximum sum, and then this process of having them come so fast that we don't get to take all the calcium out, so we get a stronger contraction, that's called summation. Tetanus would be maximum summation. That means that the signal's coming so fast that we're maximally flooded the interior with calcium. Yes, some is still being recovered, but this gives us our maximum force of contraction out of a muscle. So there's two dials that our body uses to control how forceful a muscle can contract. One is how frequently are the signals coming from the brain. The more frequent, the more summation we get. So the more force we get out of each individual fiber. We're talking about individual fibers, though. The other dial is how many muscle fibers do we include. See, we can speed up the frequency and we can include more muscle fibers. So we can really fine-tune the force that muscle can generate. All right, tetanus is what we call maximum summation. That's all that is. So now we're talking about the idea of motor units. And what that is each neuron that comes down to a muscle, that neuron's going to split and it's going to send little ends, connections, neuromuscular junctions with multiple muscle fibers. And each group of muscle fibers that get a signal from a single neuron, that's called a motor unit, because all those fibers will contract as a unit because the same signal is going to stimulate them all at once. Okay, so that's a motor unit. Now, depending on your body part, like my fingers, I have such real good, fine, precise control of because I have more motor units there. See, each one of those motor units is coming all the way up into the brain to a specific part in the brain. And the more motor units that I have, the more brain it'll take to run them, right? So it would make sense then that the things that I get the most control over get more brain control. The reason they do is because they have more motor units. And each motor unit might be composed of fewer muscle fibers than in a muscle like my thigh, where I don't have superfine control. So I won't have as many motor units in my thigh, but I'll have more fibers controlled by each one, right? So each neuron's going to many fibers. Just put it like in my hand. I've got more motor units in my hand than I do my thigh. Well, I don't have as many muscle fibers here. That must mean that fewer muscle fibers get controlled by each unit, right? And so then what we can do is we can recruit. So that's what your body does. It sends signals to more motor units as we need more force. So, and it does it in real time. So if you start lifting something, your body automatically does two things. It automatically starts causing more motor units to fire So more groups of fibers are involved. And at the same time, it adjusts the frequency of stimulation to get exactly the right amount of force. Anyway, that's kind of, look at this. You might have to rewind it a few times. Okay. Good. Okay, and this, I don't think we need to even go over. So the rest of this you can get, the types of movements and how muscles produce action and all that. And then the actual muscles, you got to get that from the lab manual and all that. The list of muscles that you need to know. And I don't show you all the pictures of that here. I will, though, post a functional. Some people like to study it that way, like functionally. I'll put up in there in your class materials a resource I use with the lower level class, but you may find it helpful. If you like it, use it. It's a packet of muscles, breaking them down by different areas. Use it as an adjunct to what you have. Okay? Cool. Alrighty then. Well, I hope you're doing well in the course and stick with it. Put your time in. You got to put time in on this. It takes a lot because there's so much stuff to learn here. But you can do it. You just got to decide to do it. All right. Let's see if I can.